Some I do have slides because I gave a journal club on this paper a while back. So we'll see what we can make of it. Did you guys read it? Yes. Okay, good. Okay, good. So in this paper, they're working on expressing recombinant spider silk in E.Coli. So I liked the paper. I thought it was fun to read, and it emphasizes sort of the problems that you'll encounter with recombinant protein expression and codon optimization, things like that. So first, the spider. So this is the spider that they're working with that they clone the genes from is Nephila clavipes. We have these around here, the banana spiders. I hadn't seen them until I finally moved to Alabama. You guys have seen these? Never?
I've seen something similar.
We have one in our yard. They make these massive webs. They're huge. So they're called the golden silk Orb-Weavers. These are like I think they talk about this somewhere in the paper that these spiders are famous for the strength of their webs and their webs. Yeah, North Carolina to Texas. So I've seen them here in Alabama in my yard.
Did you know they ate like birds and cats?
Why? That's what I read. I don't know. They must be like maybe in like the Everglades or something. I don't know. The ones I've seen don't seem big enough to eat a bird or a bat. But
actually, I do think I've seen one for one of my jobs. We were out sampling for like these little fish. And
okay.
They just said it was an orb spider. I don't know.
Yeah. Yep.
Okay.
That's an orb spider.
They actually threw a little fish up in the web.
In the web?
And he ate it.
Oh, that's awesome. Oh yeah. Okay. So here's a picture of one winding up a bat. Okay. So in theory, right, like there's all kinds of, this might be kind of like pie in the sky, but in theory, the spider silk, they talk about this in the paper. It's got all these crazy properties. It's 3 times stronger than Kevlar. So Kevlar is what we use to make like the bulletproof vests and stuff like that. So in theory, it has all these like crazy uses that we could use it for all these mechanical engineering things. And it's also biodegradable and biocompatible. So that means it talks about in the paper, one of the uses for this might be that they could use it to like encapsulate certain bio implants that you might want the capsule to degrade, or because it's made out of protein, right? It's going to be, it's not going to sit around for forever, and it can be degraded by standard like proteases and things like that. So you could use this in biomechanical engineering stuff, implants as well. So that's kind of like one of the interesting aspects of this is that it's organic. They talked about this, so that one of the questions, like, why can't you just collect this from the spiders? Well, you can't really farm the spiders because you can't grow them in a way, and because they're territorialistic and they're aggressive, they will attack other spiders. So you can't like put a bunch of these like in a small area, like in terms that you would think of a farm. And I've said this before, that there's other companies that you can grow and farm silkworms, and people have been doing this in China for like thousands of years, where they farm the silkworms and then they make the pupal casing, which is made out of silk, and then they'll unspin the silk from the pupal casing and they can use those threads to weave clothes. So people have been doing that in China for a while. And now there's people that are taking and replacing the silk genes from the silkworm and replacing them with the spider silk genes, so that the silkworms will essentially lay spider silk instead of the standard silk. Or, like this paper does, you could stick it in E. coli, which is what they try to do. So if you look at the spider silk protein, so now you guys know, like if you see a reference, like in the first paragraph, it says MASP1, which stands for the major, spindlin or spidroin protein 1, or MASP2. So there's 2 paralogs of these in these spiders. So there's 2 genes that are essentially the same thing, that encode essentially the same protein. And if you ever wanted to see these, you can just look them up on NCBI. So like I found one of these things on NCBI and I put it here, and you can look at it and just look at the sequence of it. It's very strange. And they talk about this in the paper and it's basically just a whole bunch of glycines. And you also see in this one, there's a whole bunch of alanines. I don't know if this is from a different spider. So all spiders have these silk proteins. And I would bet that they're, for the most part, the same.
The paper had mentioned that the one species, like, along with that glycine, there's just a lot of those, like, alanine sequences.
Yeah. So this is not what a standard protein would look like, where you see this is what's called like a repeat protein. And repeat proteins are, I should actually set up a new page here. Repeat proteins are where you have like a sequence. Let's say it's like G, 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 A. And you make like a polymer of this sequence to the N times. That would be a repeat protein. And the paper talks about how these spider silk proteins are essentially that. So in here, I've highlighted in green one of these repeats that has a bunch of glycines, prolines, and glutamines. And so this protein is basically just a bunch of these repeats. Okay, so the other thing with these proteins is they're massive. So because they're repeats, they literally just string these things together into very, very, very long proteins. So 250 kilodaltons, that's a massive protein. In terms of like, if this is the whole sequence, how many amino acids is this? I don't know how many amino acids, but it's this times 3 nucleotides. So if you were to put this into a plasmid, you're already going to start to get problems because it's going to be a massive gene. It's going to be in thousands, multiple thousands of base pairs long. So that's where you're going to also start to get to problems. And it's very, very hard to synthesize through recombinant protein expression stuff that's this big. And the data in the paper clearly like shows this. That's why one of the reasons I like this paper.
So with a big repeating gene like this,
Yeah.
doesn't it take up a lot of space to just repeat the same thing over and over instead of having some kind of method where it tells the polymerase, like, jump back to this point, right, jump back, right?
Yeah, in theory, what you're saying is correct. But in practice, eukaryotes have all kinds of, that's one of the major differences between eukaryotes and prokaryotes, where eukaryotes have all kinds of quote unquote junk DNA. They don't really care. They do care a little bit, but they don't really care as much as the bacteria how much extra genome they have. Because think about it like the bacteria, those are the really ones that really encounter like the time constraint on their reproduction. In eukaryotes, everything is kind of like slowed down, and so they're not under really a time constraint. They don't have as much of a selective pressure to reduce or streamline their genomes. So having like a big protein like this isn't really an issue in eukaryotes. And even like there's a lot of eukaryotic genes that are big. It's not like super uber rare. And it might actually be, so think about, this is actually a good question to talk about. So let's say you could engineer some kind of a way where like the RNA polymerase could grab this and then like come back and then like somehow like make something like this where it makes like some kind of like a repeat transcript. You're going to have to program that ability into the RNA polymerase, and programming that ability is also going to take multiple like new genes to reprogram. You know what I mean? So you're going to be also adding genes to allow that functionality, even if I don't know if that's even possible, but if it was, you'd have to do that by programming other machines. And these themselves would have a certain length, right? So you might defeat your own purpose by programming that. You know what I'm saying? But it's not an issue to have extra DNA in eukaryotes. Let's see. They talk about this in the paper that evolution has favored these big proteins. So did you guys catch why? So you could just make a protein that's one monomer. And then you could covalently link these monomers together. That would be one way. To kind of do exactly what you were saying, but in an easier way. Rather than making like a long transcript, you could just code a peptide, and then you could make some kind of a post-translational modification to link those peptides. And they actually talk about this. You can do this. And it doesn't need a post-translational modification. You could do it with interactions, like protein interactions, almost like actin. And you could like arrange a polymer of these monomeric units. But they talk about why this is not as good. Did you guys catch that?
There's something about the weight of it, or
So they say specifically that evolution has favored not this, but a singular long protein encoded by these units. And they say the reason for this is because this is fine, but these are weak. These are weaker. So if you imagine a spider needs to put pressure on each end, right? And if it's a silk strand, it's going to be pulling these proteins, right? And this, however this attaches, is going to be much weaker than an actual peptide bond, which would be in this case. So they actually say that the reason that this doesn't happen is because simply that it's a lot stronger. The tensile strength, when you pull it, is a lot stronger when it's one synthetic fused protein with these peptide bonds. So they talk about that. And then what you have is these long things, and at the ends they are like, I don't know if I'd say ionically, but they are like non-covalently like interacting with each other. But the longer they are, the longer they can span out that interaction, and so the stronger it will get. So that's why evolution has favored the lengthening of these proteins. Okay, so let's look at, here's the first figure in the paper. So they talk about how they built these monomers. Did you catch, or these different length polymers? Did you catch how they did that? Can you describe that more? Yeah, so it's in these restriction enzymes. Very good. So they can just cut this out, and if you have a bunch of plasmids, they can cut it out, and then they can religate it back together to get these different units. Because you can get a piece of this that can ligate onto here, and then you get two, and then another one can ligate here, and you can get two. So all they did is they cloned this first monomer of this, okay? And then they are cutting it up and then refusing it back together in lengthening strands. I didn't quite understand the precise the wording that you said when they wrote that. I read that and I didn't quite understand that. We could check. Let's check the NHE. Remember what this is, NHE1 and SPE1. Let's check to see what those are. Okay, so here I go into a long tangent where I'm checking the restriction enzymes, and I didn't figure it out at the time. But to correct my error, what I discovered was that the NHE1 and the SPE1 restriction enzymes, they're not the same site, but they share the same overlap. So by using these enzymes to perform the restriction digest, they can essentially just digest a bunch of plasmids together, and then when they religate, the inserts share that overhanging flap, and so they can just synthesize immediately a bunch of polymers of different sizes because of those overlaps when they religate. Okay, I didn't quite understand that then. But conceptually what they were doing is they were cutting this thing out and pasting it after the other to get these different monomers. But I guess so what confused you also confused me. So we're on the same page. But they were able to synthesize these different length mers, okay? And they tell you the characteristics, the content, the glycine content, the molecular weight. And then their hypothesis is that bigger proteins are going to have better properties. And so they test that. So this is the first actual expression. And they're just showing you that as you express bigger and bigger and bigger proteins, the amount that you can purify goes down significantly. So like here is if you just do the 32 polymer, 48, 68, and each time you get less and less. And so if you were actually going to make clothing from something of the 96 mer, you're never, ever going to be able to purify enough of that protein to make clothing. So that's like one of the big problems, is the thing that doesn't work very well is the thing that expresses the best. So they wanted to try to figure out a way to solve this problem. How can we increase the efficiency of the construction of these larger and larger polymer proteins? Skip that plasma copy. Oh yeah, and then this was in the supplemental. So they basically are looking at. They realize that this is an issue, so they want to solve this issue. Okay, so their strategy is, well, let's turn the proteins on versus off, and let's look at which genes the E. coli upregulate when it's on. Okay, and then whatever E. coli is naturally upregulating suggests what it needs when it's expressing these proteins. So they do these experiments. This is like metabolic analysis, and then the actual figure that you see in the paper, which they report as the most significant data, is they see an upregulation of Gly-S and Gly-A. So what is this type of data? Have you guys ever seen this type of data before? These dots? Did you catch what it was?
Wait, unless it's not 2D NMR, is it?
It's not 2D NMR, but it's 2D something. So you're close.
So is it 2D electrophoresis?
Yeah, it's 2D page. 2D page. That just means it's two-dimensional page. So usually what they'll do is you will make a little tube, and it's a little glass tube, and you fill it with polyacrylamide gel. So you fill it with a normal standard gel, and then they run this first electrophoresis. So they'll put the current up here, and they'll run the first electrophoresis basically based on pH. It's a specialized system where proteins will separate out based on where they like to be in a pH range. Okay, so you get an initial separation of your loaded samples, and then you can put some pressure on the tube and you pop out this little like spaghetti like gel, and you lay that on top of a standard SDS page, which is now separating based on size. And now you separate those proteins that have already been separated based on pH according to size. And what this produces is these dots. Instead of the bands that you're used to, you basically get these dots. So the first dimension is based on pH separation. The second dimension is based on size separation. And that produces, I don't know what happened. That produces these big gels that show you these dots. This is not as popular anymore. It's kind of a lot of people did this like 10, 15 years ago. It's not as popular, but you'll see it if you read these old papers. And it's a way to just get more separation. And then what they can do is they have different treatments. So here's the control, which is just a standard E. coli. Here's when they're turning on expression of the 32-mer, and here's when they're turning on expression of the 96-mer, okay? And you can see that this dot that they tell you, that they circle, gets bigger and bigger, which means it's expressing more of this protein. Does that make sense? And this one gets bigger and bigger. And they can cut out these dots so you can analyze and figure out what they are. And they're telling you that they did that. They figured out that this one is Gly-A and Gly-S. So these were two enzymes in a synthesis pathway of what? Glycine. So it's basically telling them, what is that telling them? What's their conclusion?
The cell wants more glycine.
Yes, the cell wants more glycine. And it's like the most obvious thing, because look what you're telling it to make. You're telling it to make this thing that is like needs a whole bunch of glycine. Okay, so that makes sense. And then they show you this little glycine pathway thing. Here's where Gly-A comes in. Where's the Gly-S? Is the Gly-S the tRNA synthesis? Oh, here we go. Yeah, so Gly-S makes the tRNA, or charges tRNA with the amino acid. So it basically preps the actual tRNAs with the glycine, and then this Gly-A is involved in glycine synthesis in the cell. So these are two key enzymes that are important for basically building proteins that have lots of glycine. So their conclusion is, well, let's boost the glycine levels of the cell. Okay, so then they actually experimentally do this. So they make this all fancy, and they say we're engineering the metabolism of the E. coli, which is like a really, really fancy way of saying, hey, we're just going to figure out ways to give it more glycine. So they put these genes in. These are genes that actually encode or the tRNA. So these will make more tRNA that will recognize glycine. So that's this lane here. And then they have a lane with Gly-A, which was, go back, Gly-A was the one that actually synthesizes glycine. And so they ask basically the question, if we have more tRNA and more glycine, will we get more of our protein? So here's the minus control, just standard amounts of what you're getting. And it's not very helpful to look at the 32 lane, because they're kind of already getting a lot of that. It's most helpful to look at the 80 and the 96 MERS. So here's the band that corresponds to the 96 MER. You're not getting any of it in the negative control, where they don't have this stuff. And if you add in the things that make the tRNAs for glycine, you get more of it, right? You see that? Very clear, very obvious, okay? And then if you add in the Gly-A, it doesn't help that much, okay? But the best combination that they got was when you mix in the 3 tRNAs plus the Gly-A. They basically say, look, this is the best combination. We got the most expression. This is the highest compared to all these other ones when you have the glycine tRNAs plus the Gly-A.
What is X2?
X2, wait, where's what?
X2, the third line.
X2, let's look at the figure legend. X2, lane 2, tet Gly VXY for overexpression of Gly. That encodes it. Oh, wait, no, that's lane 2. Wait.
It has two.
Wait, that's lane 3. I'm sorry.
It has 2 of the expression sets.
Gly 2. Yeah, okay. So it's basically
Oh.
the same thing as this, but times. Oh, okay. That's why. Yeah, I just made that connection that you made too, times 2. Okay. So it's like, oh, let's just give it like double the amount. They didn't try quadruple. Okay. So we got that. So they're engineering here the metabolism to be specifically designed to make this protein. Which figure was this? This is figure 3. Okay, so here's their strategy for how they're gonna purify these proteins. Because if you're gonna make and synthesize this, you have to be able to pull it out of the cell. So they do this strategy where they're not doing an affinity tag. They're not pulling it down with like an affinity. They're just doing classical biochemistry. So if you know the properties of a particular protein, you can do different treatments that cause other things to precipitate out, or other things will stay in solution. And if you know the properties of your protein, you can get actually very, very purified things by just utilizing that biochemistry. So basically, they've discovered that these proteins that they want to analyze are acid soluble. So if you treat the cells with acid, a lot of times it will cause most of the proteins to just precipitate out of solution. And in this case, so most of the proteins are precipitating out, but their proteins that they care about will stay in solution. Okay, so they do these various biochemical purifications, and here they're running an SDS page. And this is basically like a gel. The whole purpose of this gel is to say, hey, look how pure our protein products are. Look how awesome this protocol works for purification. Because you just see this is good because it correlates with the lab we just talked about. All you see are basically the bands that they want to get. There's not really anything else in there. And here is a Western blot. So what's the purpose of this blot?
Visualize things that are in very small concentration.
Not necessarily in this case, because they're not so small concentrated. They can see it with the standard SDS page in Kumasi. This is Kumasi. If you see blue, Kumasi is just a stain that binds and proteins and stains them blue. So they don't really need to do a Western blot if they're just looking for detection, because they can detect it with Kumasi. What's the real reason for the Western blot? Yes, it confirms that these are the spider proteins. That's the real reason they're doing that. Even though it's kind of like redundant, because they know the size of the proteins and they're at the perfect size. So that in that sense, that's already kind of a strong case. But you'll see this a lot in papers that they build up what's called convergence, where you do many, many, many experiments that all tell you the same result. So it's as if like a reviewer was like, well, I don't believe that. Then you're like, well, you better believe A, B, C, and D then. So they're doing a Western blot. That's correct, to confirm that these are the spider proteins. And then they look at different, is this the different media? So production, here they're looking at time. So time of what, induction? Or here's the induction. Silk production titer. Oh, what's the difference? Is this in the supplemental or is this in, this is a supplemental. Skip the supplemental. Okay, so then they make the things to test them, the fibers, and then they actually do the tests of the tensile strength to prove their hypothesis. So their hypothesis was that the longer the protein is, the better properties it's going to have. And they purify their protein. So here's the 16 mer, which is the blue. Here's the 32 mer, which is the red. 64 mer, which is the green, and then 96, which is the purple. And here they're just measuring if you pull these things, when do they snap? How much pressure can you put on them before they snap? And the figure is just very clear. It's just very obvious. As you go up in the y-axis, they can tolerate more and more stress. So obviously, these ones, the purple ones, the 96 mers are the best. Okay, so that verifies their hypothesis. I didn't have any disagreements with this data. That's what I'm saying here. Okay.
So
yeah,
how do they spin the silk? Like once you get it out, would it be stronger if you like spun it or braided it?
Well, they did somehow spin it because they got these fibers in figure 4. So they somehow got it into the fibers. Let's look at the methods. Did you guys catch that?
They did go through it. They said that they were spun and drawn under the same experimental conditions, but I was like, what experimental conditions? Because I don't feel like they really went over that.
Yeah, so here they have a whole section in the methods on fiber spinning and mechanical testing. Let's just walk through this and see if we can answer those questions. So they first say they are taking lyophilized silk proteins. What's that mean? What does that mean?
They've been treated to have an affinity for I don't know what lyo is, but affinity for something that lyo stands for.
You'll see this word a lot. Lyophilized is basically just like they concentrated by drying it. So they had a solution of some buffer that had their purified spider silk proteins in there, and then they just let all the water evaporate. So then what you'd get at the bottom of that tube after evaporation are some crystals of your spider protein. That's what lyophilized.
So is lyophilized specifically drying in that method, kind of air drying?
Yeah, pretty much. Usually they'll put stuff in a vacuum, and then the vacuum will suck out all the air, and that will enhance the drying process. So that's the first thing. So they take that. Dope solutions are extruded with. Okay. Hang on. So they're saying they dissolved the lyophilized. So they dissolved these crystals in HFIP, and then they say sigma. I'm assuming this is some kind of a buffer. And if we went to the Sigma website, we could look it up. So they dissolved the crystals in this buffer, and then it says they loaded them into a syringe. So they took a syringe, okay, and they sucked up this spider crap. I don't know, they did something in here to make them coagulate, which means they're like starting to like bind each other. You can do this with DNA. So like if you have a tube full of DNA and you take a glass rod and you stick it in there and you actually just spin the glass rod, it will like collect DNA in like coagulates, and then you pull that out. And if you pull that out, you'll actually get like a snot slob of DNA. So I'm assuming they did that in the syringe. And then they must have somehow shot them out of the syringe or something, and then laid them in a line, and then just let them dry, is what they said. And that's what would somehow produce these things. Okay. And then they did the structural analysis of these things to sort of try to figure out why the bigger ones are better. And here they see that the properties of the smaller ones, there's big holes in the fibers. And in the 60-formers, it's kind of more of like a perfect crystalline structure with less impurities. There's still some right there. But if you look at this, there aren't these like massive voids. So they make the argument that the bigger ones just kind of fit together better.
Yeah, why is 2D electrophoresis falling so kind of by the wayside?
One of the it's a huge pain. So when you make I've done it before. When you make these things are a huge pain because you have to take a syringe and stick it on to get the page shell thing out of the gel glass tip. You got to take a syringe and you have to like hold it and you have to like squeeze it, and it takes a lot of force to like push out these like gels, and then you have to take that without snapping it. So it's actually kind of hard. Like you have to be very, very gentle with it, and then you have to lay it on top of the gel, and there can't be any air bubbles. And one of the hardest parts is when you lay it on, it'll go like this. And you obviously can't do that because then if there were proteins here, they won't get into your page shell. They'll just electrophoresis into the air. So it's a huge pain in the butt to get this perfectly laid flat. Okay. And it's like they used to do it as like a semi-quantitative way, like where you would do it with treatment 1, treatment 2. It's a good question. I like this question. And then you could compare the dots and you say, oh, this dot got a lot bigger, therefore it's overexpressed. We have a lot better ways to quantitatively analyze protein upregulation or downregulation than 2D page. So nobody does it anymore because it's a pain in the butt and we have better ways. So the better ways we have now are like called iTrack. That's one. This is isobaric tagging, isobaric tagging, something quantitative, something. It stands for a bunch. You could Wikipedia it. But the premise is you take sample 1, sample 2. So it's same thing you're comparing treatments, 1 versus 2. And you want to know if in 2, which proteins are upregulated and which proteins are downregulated. So you do protein extractions, protein, and then you add basically tags, which will covalently attach to your proteins, to all proteins uniformly, okay? And then you can run this through LC-MS, and you can quantify very, very specific proteins, how much of protein X is in sample 1, and then you can compare that to how much of protein X is in sample 2 based on the number of these tags that the thing detects. And the tags are attached to peptides, so that's how you can trace the tag to a particular protein X. And then you get an Excel file that is just a list of a bunch of proteins, and they tell you by how much your sample is upregulated in 2 versus 1. And then you can just look through this quickly, rather than having to look at different dots visually, and like it's kind of qualitative because you're like, oh, I think that dot's a little bit bigger. And you like cut it out, and then you have to do the LC-MS anyway to verify what that dot is. So it's just like it's been superseded by much better technologies. And you can do this you could do an experiment like this for less than or equal to $5,000.
Oh, that's a lot less than
And this one actually, it's really what's going to cost a lot is the labor. So you're going to have to pay a graduate student for months to try to work out the details of getting data that is actually interpretable. So it's just people don't do it anymore because it's almost impossible. Yeah. Any other questions? That was a good question. What year is this? What did you say?
2010.
2000. So this is already 9 years old.